a cell are expressed to perform a particular function or a set of functions. Now suppose we have eaten suppose gulab jamun, and at that time we need more of we have taken more of the sugar is more in our body. So who is the which is the hormone which is regulating that? Insulin is the hormone which is regulating that. So if more of sugar is there, insulin is going to regulate that. We have eaten that gulab jamun, we have taken more of sugar, so insulin will be produced to regulate decrease the level of sugar in our body. Now after suppose 6 hours, the level of sugar has gone down in your body, so now the insulin need not be insulin is not needed for our body to this reduce the level of sugar. So in the case when more of sugar is there, insulin is needed, and in the case where less of sugar is there, insulin is not needed. So we should do something so that insulin is not expressed at the stage when sugar is not there in our body. So that is what is called regulation of gene expression. So it is the metabolic, physiological, or environmental conditions that regulate the expression of genes. So these are the different levels I have just explained, that is shown in a diagram. Genes on DNA transcription control, that is at the level of transcription when gene expression can be regulated. At the level of processing, splicing, this is the splicing technique. This stage also processing gene expression can be controlled. Then in this stage when mRNA is traveling from nucleus to cytoplasm, at that stage also it can be controlled. And in the last stage when translation is taking place, at that stage also gene expression can be regulated. So in prokaryotes, control of the rate of transcriptional initiation is the predominant site for control of gene expression. Now see, in prokaryotes we have seen there is no difference between cytoplasm and nucleus, and also this control of gene expression is very dominant there. Regulation of gene expression is more dominant. In a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins, which can act both positively and negatively. This sentence you will not understand now. I will explain you and then you will understand. So look here. We have seen, I'll come to this diagram later. Now first we will come to this diagram which we had studied in the previous class. We had studied, no? See, you have studied in your previous class promoter transcription unit पढ़ा था ना, promoter, structural gene and terminator. In transcription unit you have studied that promoter, structural gene and terminator. This much you have studied. Now in between the promoter and where we have studied RNA polymerase will come and act on promoter and it will help in the formation of mRNA. That is there in transcription what you have studied. Now when regulation is done, what is happening? There is an area in the gene. This is a DNA, a gene sequence in DNA we are talking about. Now here promoter is there. This is structural gene. We have studied, no? You remember, no? Promoter, structural gene and terminator. Now in between the promoter and structural gene there is a place which is called operator. So a new area also you are studying. In between the promoter and structural gene there is an area which is called operator. Okay, now this operator, now what is the function of this operator? We will come to that. Before that you come to this area. This area there is a regulatory gene. This area we call that regulatory gene, and this regulatory gene will produce regulatory protein. Okay, this regulatory gene will produce regulatory protein, and this regulatory protein will produce repressor. What will happen? This RNA polymerase, this is RNA polymerase, it will come and attach to promoter. It will come and attach to promoter and it will do the mRNA sequence. That is a normal case. Now suppose this regulatory protein is producing a repressor. Repressor means it is trying to stop this function to continue. So what happened? This regulatory protein will come and attach in this operator region. So what will happen? This RNA polymerase cannot do its work. It will be blocked. This area will be blocked. So this RNA polymerase will not be able to continue its work because this is blocked by a regulatory protein. So the coding, this mRNA formation will not take place and the gene expression would be stopped. This is the basic thing that happens in regulation and in gene expression, regulation of gene expression. I repeat, we have seen promoter, we have seen terminator and structural gene. The middle portion is the structural gene. Now, in between the promoter and structural gene, there is an area which is called operator. Now, RNA polymerase attaches to the promoter and it continues its work and mRNA formation take place in a normal case. Okay, but when this RNA polymerase is here, now where there is an area which is called regulatory gene, and this regulatory gene will produce regulatory protein which is actually repressing something. So this is it's actually repressor. So this regulatory protein or repressor, it will come and attach to this area which is the operator. So when it is attaching to the operator area, this RNA polymerase, there is a blockage here. So this RNA polymerase will not be able to form mRNA. So the gene expression would be stopped. So that is what is called regulation of gene expression. Okay, is this much clear, children? If this much is clear, we can go to the next step. If this is clear, clear, no? Okay, so this whole thing, that is what is called operon. Okay, there is a structural gene, there is an operator, there is promoter. This is called operon. Now we will study. See this. Now, when we this is an operon when we are talking about. As I told you, this is the structural gene. This is the operator. This is the promoter. Okay, and this is the regulatory gene. What I explained you, that same thing is drawn here. This is a structural gene, operator, promoter, regulatory gene. This RNA polymerase will come and bind to the promoter and it will continue mRNA formation. But if this regulatory gene is producing regulatory protein and which in turn is producing repressor, if this repressor goes and attaches to the operator, RNA polymerase will not be able to code here, will not be able to form here, and thereby protein formation would be stopped. Okay, now if this repressor is not there, RNA polymerase will be able to form mRNA and then translation and then protein will take place. These steps would take place if repressor is not there. This is the general thing what happens in an operon. Okay, so see this now. So that is what is explained here. Now, as I told you, operator. Operator, what is operator? This one. Operator, it is the sequence which interact with proteins. Which protein? This protein I told you, no, regulatory protein. So it is a sequence which interact with proteins to regulate the accessibility of promoter gene. So this is the promoter gene. The operator will combine with regulatory protein and it will regulate the function of this promoter. Okay, so they are adjacent to promoter elements. They are next to this promoter element. Then the sequence of operons bind a repressor protein. So here, as I told you, regulatory protein, it will give rise to a repressor which will bind to the operator. Each operon has specific repressor. For example, lac operon. Now we are going to study about this lac operon. Lac operon is lactose operon. We will be studying in this. There are many other types of operon, but in this your syllabus only lac operon is there, and it interacts specifically with lac repressor. Lac repressor, that is the lactose repressor. How it is, we will be seeing now. Okay, so just that this is the theoretical portion. What is operon? Operon is a set of many structural genes. You can see here structural genes arranged under a common promoter. This is a common promoter and a common operator. Okay, that is an operon, a set of many structural genes. This structural gene, this area, arranged under a common promoter and a common operator. Now, you tell me, we have studied about monocistron and polycistron. So, what is this? Polycistron and monocistron, we had seen when more proteins are formed, that is polycistron, only one protein is formed, that is monocistron. Which example is this? Is this monocistron or polycistron? Is this monocistron? Huh? Polycistron, because many proteins are formed. And polycistron, we have seen it happens in eukaryotes or prokaryotes. Prokaryotes. Okay, just remember PP, polycistron, prokaryotes. So this we are going to study in terms of prokaryotes. Okay, in terms of bacteria we are going to study. So operator is a sequence on DNA which interacts with regulatory proteins to control accessibility of promoter region. This is what I already explained you. Now, the structure of an operon, as just now I told you, an operon consists of promoter. This is the promoter. It initiates transcription. So promoter will initiate transcription. It allows RNA polymerase to bind with it. As I told you, this RNA polymerase will bind to promoter. Then it decides which gene to participate in mRNA synthesis. Okay, this is what the promoter, the use of promoter is. Operator, it is a segment between promoter and structural gene. Operator, it is the structure between promoter and structural gene. And it regulatory proteins bind here. I told you, no, regulatory proteins which are produced by regulatory gene that will come and bind to the operator. Then it is capable of obstructing the function of RNA polymerase. As I told you, RNA polymerase will not be able to work if the regulatory protein will come and attach to the operator. So regulatory proteins obstruct RNA polymerase to bind with promoter, because of which process of transcription will not be initiated. Whatever I explained you, the same thing is described here. Next is the structural gene. This part, that is the structural gene. These are the genes that code for mRNA. Then they are regulated by operon. Okay, so these structural genes are the genes that code for mRNA. So these are the main three parts. Promoter, operator, structural gene are the main three parts of operon. Apart from this, you have regulatory gene. Regulatory gene, that is this one. Regulatory gene will produce what? Regulatory gene will produce, very good, regulatory protein. Regulatory protein will go and attach to operator, and so RNA polymerase will not be able to do the work. Okay, this much is clear. If this is clear, then only when we go to lac operon, that would be clear. Is this clear to everyone? Okay, so we will go to the lac operon. Before that, regulatory proteins can be activator or repressor. Activator means they will act positively. So they will act positively. That means they will allow the RNA polymerase to work. So that will be positive regulator. And negative regulation, that means they are repressor. As I told you, no, repressor, they will try to regulate. They will try to obstruct the flow of RNA polymerase. So what we are going to study is lac operon, and that is a repressor. So that is negative regulation. Lac operon, what we are going to study is repressor, and that is why we are calling it negative regulation. That means it will stop the flow of RNA polymerase. Listen very carefully, children. This is a very, very important topic. If a lac operon, see this chapter, no, transcription, translation, replication, lac operon, and this one, DNA fingerprinting. These are very important topics of this chapter. You should know this. Pakka exam में इसमें से questions तो पक्का ही है. So you be very attentive now when we are doing this topic. Okay. So lac operon, it is an example of negative regulation. Like in your NEET exam and all, you may get this question. An example, you may be given options, and which of the following is an example of negative regulation? Lac operon. And there is a tryptophan operon, that is an example of positive regulation. Okay, but that is not there in your textbook. Just I'm giving you, if suppose it comes in your NEET exam and all, that is tryptophan operon, TRP. It's written as TRP. So who was the one who did this lac operon? Francis Jacob and Jackie Monod. Francis Jacob and Jackie Monod. See the scientist name and all, it may be helpful for you in your NEET exam. Okay, so if you remember that, that is good. So if you remember Jacob and Monod, that is enough. Okay, so they gave the lac operon. Now lac operon was of E. coli was the first operon to be discovered. Again, this can be given for 1 mark or MCQ. Lac operon of E. coli was the first operon to be discovered, and it is named after lactose operon. Lac operon is actually for lactose operon. Now, this regulation of gene which codes for 3 enzymes involved in lactose fermentation. This I will explain you. Before that, you first see the parts of lac operon. The parts of lac operon are, as we have seen here, same way the parts of lac operon would be regulatory gene, structural gene, promoter and operator. The same thing what we have seen here, regulatory gene, structural gene, promoter and operator. See children, you should remember this sequence. Regulatory gene. Now regulatory gene in your books it is written small I. It is represented by small I. Okay, because it is inhibiting, no, so that's why small I. So you should remember this sequence. अगर operator इधर कर देंगे तो पूरा वो sequence खराब हो जाएगा. So you should remember this is regulatory gene, this is promoter, operator, structural gene, POS, RPOS. ये sequence पता होना चाहिए. Operator is always between promoter and structural gene. This you should know. ओके, प्रमोटर यू हैव ऑलरेडी स्टडीड, स्ट्रक्चरल जीन यू हैव ऑलरेडी स्टडीड, उसके बीच में ऑपरेटर को घुसाना है. सो दैट इज ऑपरेटर, प्रमोटर, ऑपरेटर, स्ट्रक्चरल जीन. और रेगुलेटरी जीन इस साइड आएगा, बिहाइंड दिस वन प्रमोटर के भी साइड में आएगा. ओके, सो रिमेंबर दैट सीक्वेंस. So lac operon, it is consists of regulatory gene, structural gene, promoter, operator. Now see this. In lac, that is lactose, a polycystron, that is what I told you earlier, polycystron, because it is coding for many proteins. Polycystronic structural gene is regulated by common promoter and regulatory gene. Okay, now see here, this is the promoter, operator. अब ये देखो structural gene, यहाँ पे lac operon में you remember there are three areas, lac Z, lac Y, lac A. Okay, lac Z, lac Y, lac A. Now this lac Z will code for beta galactose gene. That is, this is the enzyme that is produced by lac Z. Now enzymes, remember enzymes are proteins. Okay, so lac Z produces beta galactose gene, lac Y permease gene, lac A transacetylase gene. So these are the three genes that are produced. These are the three proteins you can say, or the enzymes that are produced by lac Z, lac Y and lac A. So lac operon में अब ये आ गया कि structural gene, there are three areas there, lac Z, lac Y and lac A. Such arrangement is very common in bacteria and is referred to as operon. Example, lac operon, tryptophan operon. This is what I said. This is positive regulation. His operon, valoperon, there are many operons. You don't have to study all that. Only lac operon is there in your textbook. So we will see that. Only you know this. If you want, you can remember this. Tryptophan is positive regulation. Lac operon is negative regulation. Remember that it is negative regulation. ओके, सो इतना समझ में आया कि स्ट्रक्चर ऑफ लैक ओपेरॉन. स्ट्रक्चर ऑफ लैक ओपेरॉन में क्या क्या आ रहा है, रेगुलेटरी जीन है, रिप्रेसर जीन, दिस इज द रेगुलेटरी रीजन, प्रमोटर, ऑपरेटर, देन स्ट्रक्चरल जीन, लैक जी, लैक वाई, लैक ए. You should have this picture in your mind, then you can easily explain it. Okay, so we will see the different areas of lac operon. As I just now explained you, regulatory gene, I gene, inhibitor. So this ये वाला area, this I gene, this is going to code for regulatory protein, which is going to code for repressor. Okay, so this is the regulatory gene. Structural gene अभी मैंने बताया, lac Z, lac Y, lac A. Lac Z forms mRNA which will code for beta galactosidase enzyme. Now its function is to ये मैं function में बताती हूँ, इसका जब हम वो उसका function देखेंगे then I'll tell you. Beta galactose, lactose is a disaccharide, so it has to be broken down into glucose and galactose, and that is the function of beta galactosidase. Lac Y, it is code for permease which will increase the permeability of the cell. Lac A, it is coding for transacetylase, beta galactosidase transacetylase. इसका function अभी तक it has not been found out much. Anyway, all the three are required for the metabolism of lactose. Now I am sure जब मैंने इतना बोला तो you may not have understood what is this and ये क्या हो रहा है, यहाँ पे breakage क्या हो रहा है, क्या हो रहा है, or anything. I'm sure you won't have understood. अब मैं इसको clearly बताती हूँ कि क्या होता है. Now I'll take this and then I'll tell you. See, we are having intestine. Inside the intestine, you are having a bacteria. Which bacteria? That is E. coli bacteria. So inside your intestine, you are having E. coli bacteria. Now you are taking in glucose. You are taking in your food. So whatever food you are taking, that is also taken by this E. coli. Okay, I'll repeat. We are talking about the intestine. इसका जो main thing है, just understanding theoretically. I'm just telling you what is happening there. This is not given in your textbook. I'm just explaining you because ये कहाँ पे क्या हो रहा है, that will not be clear if you don't understand this. So in your intestine there is a bacteria which is called E. coli. Now this E. coli will eat whatever you are taking, that is taken by this E. coli. Now what happens? Suppose you have taken enough glucose. Okay, suppose you have taken enough glucose in your food. So this glucose is taken by this E. coli. That glucose will be taken by the E. coli. Now suppose there is less of glucose and your body is having lactose in it. Now from where do you get lactose? Any idea from where can you get lactose? How can you get lactose in your curd? Okay, curd, milk, है ना, उसमें from that you will get lactose. Now suppose you are having glucose is there in your body, this E. coli is taking glucose. Now suppose this glucose is not there and only lactose is there in your body. Okay, so what happens? What we are studying, the Lacoper and all, that is what is happening inside the this bacteria. Okay, so what happens? This E. coli has to take lactose. When glucose is not there, this E. coli has to take lactose. Now this is the place, this glucose, taking of glucose is normally what happens, and this lactose is very less. Around most of the time glucose is taken by E. coli, but only few situations are there when lactose is taken by E. coli. So in the normal situation when glucose is taken, I talked about this, no? I talked about this structural and these structural gene LAC-Z, LAC-Y, LAC-A. In the normal situation, when glucose is taken, there was no need of LAC-Z, LAC-Y, LAC-A is not needed. That process should not take place when the normal situation when glucose is there, when E. coli is taking glucose, because glucose you can no, it is the simplest form of sugar. It can be taken by the bacteria, it can be taken by our body. So in that case, no use of this lactose. So in that case, the lacoperon, that is, it should be stopped. Okay, I repeat, the normal case when glucose is needed by our body, when glucose is taken by our body, we are talking about bacteria, it will take glucose. In that case, lactose is not needed. So in that case, that we have seen that situation, no, lacoperon. In that case, lacoperon should be stopped. Okay, why? Because lactose is not taken by the bacteria. Now suppose glucose is not there, you have taken milk, you have taken curd, and glucose is not there in the body. In that case, this bacteria has to take lactose. So in that case, the structure we have seen, no, lactose has to be taken by the lactose has to, that lactose process has to go on, lac-Z, lac-Y, lac-A, all that process has to go on. Now that is what I told, lac-Z, it will produce an enzyme beta galactosidase. I told you, no, what is the function of that? This lactose has to be broken down into glucose and galactosidase. Okay, so this lactose has to be broken down into glucose and galactosidase. And which is the enzyme that is helping that? I told you, LAC-Z में जो enzyme था, yes, beta galactosidase. LAC-Z में मैंने बताया था ना enzyme, ये वाला. So ये वाला enzyme है which will help to break down lactose into glucose and galactosidase. So वही मैंने बताया, जब lactose आ गया तो ये वाला process continue होना चाहिए. When lactose is not there, when glucose is sufficiently there, then this process has to be stopped. So there should be some switch or something which is stopping, or it is when lactose is not there, at that time it is this is stopping. When glucose is there, this process should stop, and when lactose is there, this process should continue. जब lactose है, remember, when lactose is present, at that time this process should continue. At that time here there should not be any repressor. तो जब वहाँ पे repressor नहीं होगा, तभी lactose will for it will be this should go on, and beta galactosidase will break down lactose into glucose and galactose. What is the function of permease enzyme? Permease enzyme क्या करेगा? Permease enzyme, it will allow the entry of this, यहाँ पे permeability बढ़ाएगा वो, so it will allow the permeability of this lactose inside the body of E. coli. So permeability will be increased by the enzyme permease. Okay, so that is the function of the second enzyme, permease. Beta galactosidase will break down lactose into glucose and galactose, and permease will break down permease will increase the permeability. This trans-acetylase gene, its function is not yet known. Okay, not yet, as such it is not yet known, just it is going to provide stability and all would be provided by this trans-acetylase gene. Its function is not yet known as such. Okay, so ये बात clear होना चाहिए कि when lactose is present, this process will go on. When lactose is not present, this process will not go on. Glucose will do its normal function, so this process should not go on. Okay, so see this. Now, this is what I told you earlier. थोड़ा बड़ा करती हूँ. See, this is a normal lacoperon. Here, this is a regulatory gene. Okay, and this is the RNA polymerase. This is operator region, LAC-Z, LAC-Y, LAC-E. Now, in a normal case, lactose, this ये regulatory protein क्या बनाएगा? Regulatory gene क्या बनाएगा? Regulatory protein, and this is the repressor. Repressor, how it is found? With the help of inducer. Inducer और repressor मिला के क्या बनेगा? Inactive repressor and inducer. That is, inducer will be what? The lactose. So if lactose is present, this is the lactose. So if lactose is present, in normal situation, this, when glucose is there, in normal situation, this will go and bind to the operator and the process would be stopped. Now, when lactose is there, when the lactose is absent, this will go and bind to this operator and the process will be stopped. Now, in this situation, when lactose is present, it will go and bind to this. So what will happen? ये देखा यहाँ पे claw जैसा, you can see a structure. So जैसा tongue होता है ना, चिमटा जैसा होता है. So when lactose is absent, it will go and hold it. जैसे चिमटा hold करता है वैसे इसको जाके hold करेगा और यहाँ पे process को stop कर देगा. And in this case, when the lactose is present, it will open it. This side it will open it and it will it will open and it will come out. So what will happen? This lacoperon procedure, it will continue. Okay, I'll show you another diagram for that. ये देखो, you just understand this diagram. I think this is given in your textbook. ये देखो, यहाँ पे lacoperon in the absence of inducer. In the absence of inducer, who is the inducer? Lactose is the inducer. So when inducer is not there, that means when lactose is not there, what happens? This repressor will go and bind to the operator. Okay, inducer and repressor, when both are there, this will not happen. So here in this case, remember, lactose is the inducer. So here lactose is absent. In the first case, lactose is absent. So what happens? This repressor will go and bind to the operator region. So इसमें lactose का कोई काम आगे कुछ होगा ही नहीं. Repressor binds to operator and prevents RNA polymerase. So RNA polymerase will not be able to come to this area. So this is the case when lactose is absent. What I told, when glucose is there in your body, sufficient glucose is there in your body. Now in the second case, when glucose is not in your body and lactose is present. So that is what is here. So repressor plus inducer. Okay, inducer क्या है? Lactose है. So repressor और inducer मिला के क्या बनाएंगे? Inactive repressor. So ये repressor अभी active है. If you talk about repressor, that is active now. That is why it is trying to stop it. और जब उसमें lactose आके मिल जाएगा, so वो repressor क्या बन जाएगा? Inactive हो जाएगा. So when it becomes inactive, it will come out. So this is inactive repressor. It will not go and bind to the operator. So यहाँ पे genes स्विच ऑफ पोजीशन में है, यहाँ पे जीन्स स्विच ऑन पोजीशन में है. So in this case, since repressor is not binding to operator, this RNA polymerase will do its function. It will come and form its mRNA. Okay, I repeat. फर्स्ट केस तुम लोग सिर्फ ये डायग्राम याद रखो. इन द फर्स्ट केस when lactose is absent, that means glucose is there in your body, sufficient glucose is there in your body. In that case there is no use of lactose. So what happens, repressor will go and combine to the operator region. When repressor will combine with the operator region, RNA polymerase will not form its